Hi students, good evening. So today we are going to have a rapid revision on the biomolecules. Okay. So I welcome all of you for this rapid revision session on biomolecules. Talking about this chapter on biomolecules, we will get around some 10 to 12 marks from this chapter. So that is why this chapter definitely we will get either a match of following or a sort of MCQ sort of question, direct question. So this chapter has got nearly around some 10 to 12 marks we can score from this chapter. So rapid revision round on biomolecule is very important. Being having a background with physics and chemistry and additional biology knowledge on this will help you to score easily also. So let us start with this rapid revision on the biomolecules. Okay. So I welcome everybody for this session. So let us move on to the questions. I welcome everybody for this session. So let us move on to the questions. So we'll start with the questioning session. Okay, let me start. So the first question is an example of protein. Just one minute. With a quaternary structure. Okay. So we'll start with the questioning session. Okay. So you can see here. So the first question is an example of protein with a quaternary structure. So here they have given either it can be myoglobin, hemoglobin, keratin and all of these. So what should be your answer? Okay, I am just waiting for your answers. Yes, children. So what is an example of a protein with a quaternary structure? So when you look into these what you call the myoglobin, hemoglobin, keratin and all of this, first you should understand what you mean by the quaternary structure. It is actually arrangement of more than one protein molecule in a multi-subunit complex. So the best example always we give the DNA polymerase or the hemoglobin as well as the ion channels. So they are all nothing but the quaternary structures. So when you take the myoglobin, actually it is something that is present in the muscle which has got that oxygen combining capability, okay, particularly for that. So here when you talk about myoglobin, it has got that functional single sequence and it will not have that multiple subunits like the hemoglobin. So it will be like an amino acid sequence, okay, of a polypeptide chain, okay. So you should understand that the myoglobin will have more or of like tertiary structure, not a quaternary structure. And talking about the keratin, okay, the keratin, it is going to be a different, like it will have a secondary structure, okay, so that is going to be different. So among this, when you take which will have the structure, yes, the answer is hemoglobin, okay. So the answer is hemoglobin, like as you all see. So when you see myoglobin, I already said it will have the tertiary structure. What about the keratin? That is those proteins, they will take a 3-dimension and have folds, right? So they will have either a, like it will be mostly alpha keratin and we have secondary structure beta pleated also, okay, but here in keratin, alpha keratin, here it is a fibrous structural protein, okay. So that is made up of amino acids which will have that repeating secondary structure. So what should be your answer? It is hemoglobin. Excellent student, Jeevita and Akshaya, very good. We'll go to the next question. Here the next question is an example of sulfur containing amino acid. So we all know that in our body there are 20 essential amino acids, and these are all the essential amino acids. So we have lysine, serine, cysteine and tyrosine. Among these amino acids, one is sulfur containing amino acid. So I just wanted you all to pick it up. Which would be the right, ma? Yes, please post it in the chat. Yes, very good. So which is the amino acid that has got the what you call the sulfur containing amino acid? Yes. Which one has got the sulfur containing amino acid? Yes, students, you are all right. Yes, it is none other than the cysteine. So you can see the lysine, okay, you can see the lysine, serine and the tyrosine. The tyrosine forms the precursor, it comes from the phenylalanine, right? So you can see that all these they don't have, but what has the sulfur? The sulfur comes from has it in the cysteine. So the sulfur containing amino acid, you can very well observe it in the cysteine. So the answer is cysteine. Okay. So very good, student. Very good. You have given the right answer. We'll move to the next question. Here there is an interesting question. Okay. So what is the interesting question? We'll all see. So here comes the lipids that insulate the nerve fiber are lecithin, cholesterol, suberin and glycolipids. So what should be your answer? We know that the nerve fiber, okay, like we saw it even in the previous session also. So they are insulated, right? So we call that as what? The myelin sheath, right? This myelin sheath is discontinuous, right? And they will be having regular interruption. They are not continuous. They will have that breakpoints which we call it as the nodes of Ranvier. All this we saw. But these sheaths, okay, they are all actually lipid. That main purpose of insulin, that is the myelin sheath, is what? Insulation. So what is the purpose and what is the lipid that it is made up of? Okay, what is the answer? Whether it is lecithin, cholesterol, suberin or glycolipids. So what should be your answer, ma? Jeevita, Akshaya? Yes, very good. What should be your answer? Yes, students? Yes, very good. So the answer is the glycolipids, because the glycolipids are the one which is present in the myelin sheath of the nerve fibers. Now you should see that the glycolipids that is present in the myelin sheath of the nerve fiber, actually it will surround the entire neuron. So the main thing is called the galactocerebrosides. So this is the glycolipid that is present. And apart from that, we have what is called the myelin basic protein, like that. Okay, so actually from the 5th month of the intrauterine life, the myelin sheath will form when we are inside our mother womb itself, which we call it as myelogenesis. And the vitamin B12 and folic acid, they are very important for the myelin sheath formation. Now coming to the other option like lecithin or suberin, they are not the one that is going to help in the myelin sheath formation. They say that lecithin can be one of the precursor for producing the acetylcholine. So somewhere that is linked with that memory, Alzheimer's, like that. But coming with suberin, you can see that those waterproof, okay, waterproof area of that plant cell wall, that is contributed by the suberin. Okay, so that we have to remember with, that is in the periderm. Okay, that is derived from that coke, what is that called, the cork, the oak. Okay, so that you can remember. So these will not fit into your option, rather it is the glycolipids. So what is your answer? The glycolipids. Okay students, very good, you have given the right answer. We'll move on to the next question. The next question is very simple, direct and very basic. We all know our genetic material, the DNA, and we know the entire structure of that. Okay, so the nucleoside is sugar plus nitrogenous base, sugar plus phosphate, nitrogenous base plus phosphate, and we have what is the purine and the pyrimidine. So what you call it as nucleoside. So if you know the very basic of it, like you can very well answer this question. Okay, so we know that we have got what is called the nitrogenous bases, the sugar and the phosphate, like they are going to form the backbone, right, of the DNA molecule. So according to the Watson and Crick, their proposed model, like how it is this arrangement. So we have what are called nucleosides and we have the nucleotides. So what is a nucleoside, ma? Yes, yes students, you are giving the right answer. Yes, very good. So students started giving the answer. So it is nothing but, the nucleoside is nothing but the sugar and the nitrogenous base. Now if sugar, nitrogenous base with the phosphate, we call that as the nucleotide. So that should be your answer. So nucleoside means what? It is only the sugar. It can be a ribose sugar or it can be a deoxyribose sugar. And we have the nitrogenous bases. What are those? Purines and pyrimidines. And if only with those purines, pyrimidines along with the ribose sugars, okay, it can be either a ribo or deoxyribo, they form the nucleoside. But if phosphate is attached, we call that as the nucleotide. Okay, you are correct. Very good, Akshaya, Jeevita. Now coming to the fifth question, in the composition of cellular mass, okay, so we have our cell, arrange the components protein, carbohydrates, lipids, nucleic acids in decreasing order of mass percentage. So you are going to arrange it in the decreasing order. So you imagine the cell in the mass, like how is these components that they are distributed. So what should be your answer here? So when you take an average living cell, the majority we have what is the water. So it will constitute say somewhere around 70 to 90%, right? And then we have what is the protein, then we have the carbohydrates, yes, the mass as we say. So the proteins will be constituting say around some 10 to 15%. Then we have the carbohydrates around some 3%. And then we will have the nucleic acids which will be around 5 to 7%. And then we will have the lipids which will be around some 1 to 2%. So this will be their order, 1 to 2%. So how will we are going to arrange them? Yes students, you are right. So we are going to arrange it in the order of, first what comes, leave about the water, we are talking about the mass. So it will be the protein, right? Then what comes? The nucleic acid, then the carbohydrates, then the lipids. So this is the mass percentage in the decreasing order that is arranged up. Okay children? Akshaya, Jeevita, you are all able to follow, ma? Yes? Yes, very good. Yes, very good. So that should be your answer. Excellent. So students, another important thing when it comes to the biomolecule chapter, actually many people they'll try to skip this chapter. But from the examination point of view, definite you have the 12 marks. At least 12 to 15 marks is distributed from this chapter. Second thing is thinking that as we know of something related with physics, chemistry, the question will be put forth. Please, it is more oriented with your biological aspect. So you have to furnish, like you have to brush up with these ideas before studying for the biomolecule concept. So with that, many students they skip and lose marks. So that should not happen. So get that practicing mind. Please do interact with this lively interactive session so that we can have a good idea about it. Excellent Jeevita, you have given the right answer. Now coming to this vitamin that promotes the synthesis of collagen fibers. So we have the biomolecule, the collagen fibers, and it helps in healing. So which of the vitamin is going to help in the synthesis of collagen? We all know that collagen, as it forms the extracellular matrix, which is also derived from that fibroblast which we saw in the previous session, and these one are very much concerned with repairing and remodeling of the tissues. So vast research work has been gone. So in this, one of the vitamin is contributing, whether it is vitamin A, K, C or E. What should be your answer, ma? Yes children? So what should be your answer? Yes, excellent. Yes, very good. Jeevita, Akshaya. Yes, I want you all to post the answers. So what should be your answer? Yes, it is none other than the vitamin C. So the vitamin C is the answer, which is very much helpful in what? Wound repair and wound healing. That is why honey has got that contribution. They say even for traditional way of treating certain ulcerated wound, we used to pour that honey, so or pack it up with it. What is the need of it? Because it is so rich with vitamin C, it has got the ability to treat the ulceration. So that much healing effect the vitamin C has got. Okay, very good students. Next see, we are going to mark the odd pair in the following. So here somewhere there is a odd pair. See, when I say like this odd pair, first you should see in what way they have linked. See here, amino acid protein, nucleotide DNA, glycerol fatty acid, monosaccharide cellulose. So here one side they have given the basic units, and other side they have given the structural molecules. So how are you going to pick out the odd pair out here? So I am waiting for your answers, children. So how is that we are going to make it up? Yes, so what should be your answer? So we all know like proteins, they have the basic unit. What is the basic structural unit of any protein? We have what is the amino acid, and we know the nucleotides, that is the deoxyribose sugar with the what we call the nitrogenous bases and phosphate, they form the nucleotide, and that is how the DNA structure. We know that spirally, they are helically, okay, that concept we know. What about the monosaccharides? That basic unit of the cellulose. So here the glycerol and fatty acid is the odd pair. Why is that? Because see here all the polymers we are making up. See, when you take monosaccharide, it is a polymer of cellulose. The amino acid a polymer of protein, but here the fatty acid, when you look through, it is not a polymer of glycerol, because when you take a fat molecule, you will have two main components. One is the glycerol, another one is the fatty acid. So they are not the polymers actually. So indeed they are the two components of a fat molecule. So when you take glycerol, it will have 3 carbon 5 hydrogen 3 hydroxyl group, but here it is a long chain of hydrocarbon with hydroxyl attached, with a carboxyl group attached. So that is how. So they are not polymers. So that is the reason, and this is the odd pair out. So glycerol and fatty acid. So that should be your answer, children. No, it should not be monosaccharide cellulose. Monosaccharides are what? Those basic units of glucose, no? So whether it is starch or cellulose, so what is the basic monosaccharide unit? So that will be the polymer. So the answer should be glycerol and fatty acid, as fatty acid is not a polymer of glycerol. Okay. So we'll go to the next question, ma. Akshay and Jeevita. Bushner discovered a complex enzyme system. So he is a pioneer there. Actually, he discovered a complex enzyme system from yeast cell. So his enzyme was, and that enzyme got a name, and he was awarded Nobel Prize for the same also. Amylase, which induces digestion of cellulose. Zymase complex, which induces synthesis of vitamins. Zymase complex, which induces the synthesis of ethanol. Catalytic RNA like ribozyme. So what should be your answer? Yes, students, I am waiting for you to post the answer. Says, come on, please put it up. Yes, unless you interact, the session may not be that interactive. Okay, it's a very nice interesting topic. Please students, please start to post. Yes, excellent, you are giving the right answer. Actually the enzyme is called the, so it's not the, see we are talking it from the yeast, so it is not the amylase or the catalytic. So the enzyme is zymase. So how are we going to say that zymase was the first, okay, it was the first isolated enzyme from the yeast, from the yeast cell, but we have to now know what is actually the zymase complex was induced while synthesizing the ethanol, not the vitamin. So that is where he was awarded. So you can see the man, the pioneer. So he won the Nobel Prize actually for what you call providing a cell-free extract of that zymase, where when he was able to ferment the sugar to produce the ethanol. So that was the discovery made by Bushner. So that is the answer. Okay, children, very good. Next question. In structure the keratin of mammalian hair. So the hair actually is a keratin. We are aware of it. So what is the structure of that keratin? Whether it is alpha helical like silk fibronin, beta pleated like silk, alpha helical unlike silk fibronin, or beta pleated unlike silk. So what should be your answer? See, silk fibronin is actually a simple amino acid, mostly with hydrocarbons as their side groups, and a result of this they will have a very strong hydrogen bonding, that is with the silk fibronin. Okay, they will have that beta pleated sheets with collagen as well as the the poly L lactic acid. So that is with the silk fibronin. So what would be the structure? Like it is just same as silk fibronin, or it is unlike the silk fibronin. So that you have to tell me. The keratin, the mammalian hair. So that is actually found in all the the nails, hair, even the hoofs of the animals, right? So all the tougher part, the keratin is so strong. So which confers that strong? It's the keratin, the structure, because of that so much of oven matter in it that gives the strength to the structure. So what is that keratin actually made up of? Yes students, you are right. It is none other than the alpha helical structure. So it is the alpha helical structure which we have, which is a secondary structure of the protein. As you can see the those three-dimensional folds that's there. And you can see that it is unlike the silk, because the silk fibronin, as I gave the answer earlier, it is as a beta pleated structure. Silk fibronin as a beta pleated structure. But what about the keratin? It is alpha helical, which is unlike the silk fibronin. It's not like silk fibronin. It is quite unlike the silk fibronin. So that should be your answer. It's alpha helical, right? But it doesn't look same like silk fibronin. Okay, so that is what, see, it is not beta pleated. This answer is not there, this answer is not there. So it should be either 1 or 3. In this, what should be your answer then? It is not the silk fibronin. So what should be your answer? It is alpha helical unlike the silk fibronin. So that should be your answer. Excellent students. Okay, so it is not beta pleated. Silk fibronin only is beta pleated. Simple amino acid, that hydrocarbons group that will be attached with strong hydrogen bonding, like beta pleated sheets will be there. Okay, so that should be your answer. Excellent Jeevita and Akshaya. Now which of the following is an anti-fertility steroid? Now we know the secondary metabolites, right? One such product is this anti-fertility steroid. We know that it is obtained from the plant in trace amounts, but it has got so much of economic importance as well as the medicinal values. So one such is the anti-fertility steroid, whether it is estradiol, digoxin, progesterone or stilbestrol. So what should be your answer, ma? I am waiting for your answers, children. The anti-fertility steroid, see, we know the steroid hormones, right? In that we are talking about anti-fertility. So anti-fertility means what? Anti-fertility steroid that will not allow you to have the conception. Okay, anti-fertility steroid. So what should be your answer? Yes students, I want you to post the answer. Excellent, you are giving the right answer. It is nothing but the digoxin. So this digoxin is obtained from the yam plant. Okay, so this is the anti-fertility steroid. See, estradiol is what? The estrogen like compound. The stilbestrol is same as the product of estrogen. The progesterone we have. These are all not the anti-fertility steroid. The anti-fertility steroid is the digoxin. Okay, so because we know the estrogen, the the progesterone, all that, they are all used for what? They are actually, those hormones help us in proliferation as well as the growth and development of the reproductive structures. But anti-fertility steroid is digoxin. Okay, so very good students, you have given the right answer. Excellent ma. Very good Jeevita, Akshaya, very good. We will go to the next very simple question. Which of the following classes of biomolecules are known to have catalytic property? So when we say catalytic property, we are talking about the enzymes and specifically the bioenzymes that is going to help in conversion of the products. Okay, they will act on a substrate, enhance the reaction, then we will have the product that is being formed. So which among the biomolecules, we are aware of the biomolecules which is there, so which has got the catalytic property, that is enhancing the reaction, whether it is protein and lipid, protein and RNA, protein and carbohydrate, carbohydrate and lipid. So what should be your answer? See, among the biomolecule itself, there should be a class of molecules, the biomolecules, that is going to help in the catalytic property. So among this, which is going to be the providing that catalytic property? Yes, students, you are giving the answer. Yes, the answer is nothing but the protein and the RNA. Yes, so the answer is protein and RNA. See, because we know those enzymes, they are protein nature, and to some extent the RNA, it cannot be either lipid or carbohydrate and lipid. So once when you narrow down your answers, okay, so what should be? It should be protein and RNA. To some extent they have the ability to provide the biocatalytic effect on various molecules. So that is why we call the molecules, the biomolecules that has got the catalytic properties as proteins and RNA. Okay. Very good, children. Yes, now here comes a very nice, interesting, and it is going to be triggering up your mind, because unless you know the entire concept of enzymatic reaction, you cannot answer this question. Okay. So what should be your answer here, ma? We will just see. Which one of the following statement is incorrect? First, you are going to pick out one incorrect statement. A competitive inhibitor reacts reversibly with enzyme to form an enzyme inhibitor complex. In competitive inhibition, the inhibitor molecule is not chemically changed by the enzyme. The competitive inhibitor does not affect the rate of breakdown of the enzyme substrate complex. The presence of competitive inhibitor decreases the Km, that is that Michaelis constant, that value, no, Km, of the enzyme for the substrate. So what is the statement that is incorrect? First, you should know about what is competitive inhibition. In competitive inhibition, what will happen? See, suppose you are having the substrate. Here for that one single site, two molecules will compete. One will be the actual enzyme that is there, another one is that inhibitor. So it will have exactly, it will be same analog as that enzyme, and it is going to compete here and it is going to bind, so that there will be no product formation. But what will happen? Suppose like this is going to stay forever? No. Suppose you are going to increase, suppose you are going to increase the substrate concentration. What will happen? That effect of that inhibitor will be changed. Okay, so that concept you should know. So what is happening? So this concept you should know. Okay, so that is how it is going to act. Now another thing, whenever the substrate concentration is increased, the inhibitor effect will get nullified, and at the same time the Km, what is Km? The substrate concentration when that is required to achieve half the Vmax, at least to achieve half the, what is Vmax? The maximum velocity of that enzymatic reaction. So what happens here? Usually what will happen? It will increase. The Km value will increase, okay, in the competitive, and what will happen to the Vmax? It will remain unchanged, because there is no product formation as the inhibitor interferes with binding with the substrate. So the affinity of enzyme to the substrate actually will increase as the Km is going to increase. So you should know that Km is proportional to that affinity. So what you are supposed to know here in this answer? It is the fourth. So what has happened? A competitive inhibitor reacts reversibly with enzyme to form enzyme inhibitor complex. Yes, they are going, it's a reversible combination, and it is going to form the enzyme inhibitor. Whenever the concentration of the substrate is increased, this is reversible. Yes, this is correct. In competitive inhibition, the inhibitor molecule is not chemically changed by the enzyme. Yes, whatever the inhibitor, the enzyme is not going to change the chemically the inhibitor molecule. Yes, this is true. What about the competitive inhibitor? Does not affect the rate of the enzyme breakdown. Yes, it will not have any change with the Vmax. So what is the next one? The presence of the competitive inhibitor decreases the Vm. That is not there. It always increases the Km of the enzyme for that substrate. So this is the incorrect answer. So the option should be what? The fourth one. Yes, students, Lakshmanan, Jeevita, Akshaya, you have given the right answer. Excellent, ma. Then we'll move to the very simplest question here. Which one of the following is connected by two hydrogen bonds? Very simple. Adenine and guanine, adenine and thymine, thymine and guanine, cytosine and guanine. If you know the Chargaff's rule, okay, then this question is a direct sweet laddu given in your hands, where you can very well just like that make the answer. What, Akshaya? What should be the answer here? Yes, that is connected by 2 hydrogen bonds. So according to our Chargaff's rule, what is happening? The A always pairs with what? T. So it will be by 2 hydrogen bonds. But what about the guanine with cytosine? It is the 3 hydrogen bonds. So what should be your answer? It is the adenine that always pairs with the thymine. And here how many bonds you are seeing here? It is the 2 hydrogen bonds. So what should be your answer? Adenine and thymine. Excellent, student. Very good. You have given the right answer. We will go to the next question. Which of the following statement is not correct? So again, see, many of the mistakes when we see as we evaluate the paper, the student by hesitate, they are very hasty. They want to make it fast because they want to finish in the time slot. They don't look at whether they want to make a right statement, pick up that one, or incorrect. So first give that importance. So read the entire question properly. So that itself is a very good habit that you need to develop when you are especially doing your MCQ question. Patience is important, concept is important when you are taking up a NEET like exam. Okay. So starch is a polymer of alpha glucose. Starch is made up of amylose and amylopectin. Amylose is linear structure consisting of several glucose residues joined by 1,4 glycosidic linkages. Amylopectin is a straight chain with several glucose residues joined only by 1,4 glycosidic linkages. Now when you talk about starch, okay, actually what is starch? It is a storage polysaccharide, as we all know that, yes. And it has got 2 main components, which is one is the amylose, another one we have what is the amylopectin. Now when I say amylose, it is actually a linear chain of those glucose, what you call the residues, which will have this 1,4 glycosidic linkages. Okay, but what about the amylopectin? Okay, it will have the branching there. It will have straight chains and branching, and at the branching points, okay, it will have what is called 1,6 glycosidic linkages. Okay, so that you should know. Okay, children. So here we will just see starch is a polymer of alpha glucose. Yes, as we saw there, the cellulose is a polymer of monosaccharides. Starch is a polymer of alpha glucose. Starch is made up of amylose and amylopectin. Yes. Amylose is a linear structure consisting of several things joined by that 1,4 glycosidic linkages. Yes. Amylopectin is a straight chain? No, it's not straight. It's a branching chain which will have several glucose, but where they are joined, it is by the 1,6 glycosidic linkages at that branching point. So this point you should know. So what is the option? So the right one which is not correct is the amylopectin. Another information which I would like to give you is the cellulose. Cellulose is also a storage type of polysaccharide. Here we will have what is the beta diglucose, okay, the polymer that and here we will have the beta glycosidic linkages. They will be linked by beta glycosidic linkages. So this is with the starch, the animal, and the cellulose, okay, with the plant. Okay, now here comes an interesting question. They have just given some diagrams, okay, and here you are going to match what are the nitrogenous bases of these diagrams pertaining with the combination that they have given. Okay, children. Very good, Akshaya, Jeevita, Lakshmanan. You have given the right answer. So A, B, C, D, E. These are the diagrams. They are representing the nitrogenous bases, and here you are going to find out what is what. Basically, we have what are called the purines and we have what are called the pyrimidines, right? Yes. So with this idea, we can very well make it. So what are those pyrimidine actually? The single ring structures. Have this in the mind. Very simple way we will target and make it. Okay. It will not involve that big chemistry and all. But what about the purines? What about the purines? The purines are the double ring. It will have 2 ring structures. Okay. So that you should know. So that you have to know the pyrimidine and the purines. So coming to the pyrimidines, okay, so you can see the A, which is the uracil, and then we have the C, the thymine, and then we have E, the cytosine. Okay, here mostly you see the uracil and the thymine, we see the single ring. Okay, but what about the other things, like you see the adenine, okay, and then you see the guanine. So here and all, what you see the double bonding there. So this double bond, the double ring like structures will indicate the what? It will indicate the adenine and guanine. So this way you can very well make it up. The double bond what you are seeing is not present here in any of these structures, but here see characteristic, you see the double bond structures. So that is also, and they will have that NH2 group, that is also characteristic which is not observed here also. Students, you are able to follow. So the answer is what? A is what? Uracil. B is adenine. What is C? The thymine. What is D? The guanine. And E is the cytosine. So these are the simple steps that you can apply, and you can find out what structure that is representing the nitrogen bases. Okay children? Yes. We'll go to the next question. Yes, very good children. Akshaya, Lakshmanan, Jeevita. Okay, you have given the right answer. See, this can be uracil. Okay, this can be uracil. B is guanine, C is thymine, but D is actually it is guanine. See, I told that double bond structures that you have to take into consideration. Okay ma? Okay, I think with this you will be able to understand the basic concept. Okay. Now coming to the 16th question, which ratio is constant for DNA of a particular species? So this is a very important MCQ where many times they have asked this question. A plus G by T plus C, or it is A plus T by G plus C, or A plus C by T plus G, or A plus U by C plus G. So what is that you should apply here? Now coming again back to our Chargaff's rule. We know that Chargaff's rule, in Chargaff's rule what is happening? We know always an adenine pairs with a thymine with 2 hydrogen bonds, and we have always a guanine with 3 hydrogen bonds will pair up with the cytosine. That concept we know, and always they have that what you call the thing is constant for any species, right? It is at any moment when you take this pairing is going to be constant for any of the pair. Yes, very good students, you have given the right answer. So what should be the answer? Now when you take this, it should be equal to 1, and here nearly approximately from 0.4 to 0.9 it should come. So mostly when you take, it will have that constancy. So that is where, so A plus T divided by G plus C will be the answer. It is not anything else. Excellent students, you have given the right answer. Now coming to the next question, which one of the following pairs of nitrogenous bases of nucleic acid is wrongly matched with the category mentioned against it? See, the questions when I just started picking up for the biomolecules, it was very basic. Actually, they do ask very basic questions, and they are just putting the concepts in various form, but when you take the base, it is only Chargaff's rule, or it is coming with the structural part or with some function part. But the students are finding it difficult and they do make mistake. So my sincere advice is look it in 360-degree angle of any question when you are preparing, so that avoid making mistakes. So you have adenine, thymine, whether they are purine, thymine, uracil, pyrimidine, uracil, cytosine, pyrimidine, and then guanine, adenine, purine. So what should be your answer here, children? So what are those purines? Okay, the purines that which I told you. Okay, the pyrimidines, actually they have that 6-membered ring, that heterocyclic ring. Okay, that base concept we know. What about here with the imidazole? We will have the purine group. So what, always that A will pair with the T and G with the C, that we know. And in DNA, and they form that backbone, the nitrogenous basis of the DNA molecule. In RNA, what happens? It is replaced by that uracil. So what should be your answer? Excellent students, you have given the answer. See, the difference that it comes between that is the heterocyclic component. Usually when you see the pyrimidine, they have that heterocyclic. You can see the heterocyclic rings. Okay, they are 6-membered ring, heterocyclic rings. In pyrimidine we have the imidazole, that is in purines we see. So here the purines, they contain the 6-membered nitrogen ring fused with an imidazole ring. But what about the pyrimidine? It is only 6-membered which have the nitrogen containing. Here it is imidazole, but here it is a nitrogen containing. So what should be your pairing, which we already know the base pairing. So here, so you see thymine, uracil, they are pyrimidines. What about uracil and cytosine? They are pyrimidine. What about guanine and adenine? They are purine. But here adenine, okay, adenine is a purine, but thymine is what? It is a pyrimidine. So this is the wrongly matched pair. Okay, so Manjula, Jeevita, Akshaya, Lakshmanan, very good. You have given the right answer. So next time how the question can be, like they can put it like this, say pyrimidine, what is that is getting the difference that you sign? Both are 6-membered nitrogen containing ring, but here in purine it will be fused with the imidazole ring. So like that the questions will be put forth. Next, coming to another question, here we have what is called the Michaelis constant, the Km value. We have the Km value of enzyme. Okay, that is the substrate concentration at which velocity of reaction is Vmax, 1/3 Vmax, half Vmax, 1/5 Vmax. So what should be your answer? Yes students, I am waiting. Already in one of the slide we discussed about Vmax and Km. So if you know that, we can very well apply here about the constant. So what is that Km which I spoke to you all about? It is nothing but the substrate concentration which is required to make or to achieve at least half the Vmax. So that should be your answer. So see, it is something that is required at least to make half the Vmax. So that is what we call it as the Michaelis constant. So what is that Vmax? It is the maximum velocity of the enzymatic reaction. So that concept also you should know. Okay. Yes, very good children, you have given the answer. Excellent. So coming to the next question. In sea urchin DNA, which is double stranded, 17% of the bases were shown to be cytosine. So they are saying that 17% is shown to be cytosine. The percentage of other 3 bases expected to be present in the DNA are what? So we have to find out. They have given only for the cytosine, and you have to find out for the other, like how they will be distributed. So again we have to apply what is the Chargaff's rule. So A is pairing with the T and G is pairing with the C. Now how much is this? The 17%, right? So here we have to know that they are expressed with a ratio of 1:1. So if cytosine is going to be 17%, what should be your other value? So if you see the guanine, yes, so how much it should be? It also should be 17%. Then what about, it cannot be 8.5, or because we know already it is given 17. So these two cannot be there. So narrowing down to here, see when it is 17 we know they are always expressed with this ratio. It cannot be 16.5 or anything. So it is 17%. So how it should be? It should be equal 33%, 33%. So what should be your value? Yes. So in DNA the complementary linkages are always A to T and G to C. So that when you take equal proportion and take sum of all these percentages, it will be equal to 100. So that is the concept that we have to apply here. So what should be your answer? Yes, in sea urchin it is 17% of guanine, 33% of adenine and 33% of thymine. So this should be your answer. Okay, very good. Manjula, Lakshmanan, Jeevita, Akshaya, they are just bombarding with answers. Excellent students, very good. As I start the question and I go before the explanation, these students are so brilliant, they give the answers. Very good, very good. So which of the following is incorrect regarding the amino acids and their functions? So somewhere there is an incorrect thing that they have given. So just we will see the answer. Okay. So tyrosine converted into epinephrine molecule and used in the synthesis of melanin pigment. Glycine involved in the formation of heme. Tryptophan helps in the synthesis of auxin hormone. Histidine can be converted into histamine by the removal of amino group. So here how are we going to make? So we know that we have 20 essential amino acids Those are the building blocks of those proteins, and we have all these molecules. Say tyrosine. Tyrosine is something like from that only the catecholamines are formed. So phenylalanine is a precursor, and from that we have all these dopamine, epinephrine, norepinephrine. Okay, the catecholamines are synthesized from the tyrosine, and even the melanin pigment, that is for the melanospore. So they are also are produced from that only. So that concept we have to know. What about, so this is something right. What about glycine? Yes, we know that what happens in the mitochondria. Okay, usually the succinyl CoA we have, right? The succinyl CoA, when it combines with the glycine, it forms the heme. So it is the one that is going to form the heme molecule. Okay, then we have the globin, the 2 alpha and 2 beta chains that forms the polypeptide chains that gives the quaternary structure of the hemoglobin. So glycine is involved in the formation of the heme. What about tryptophan? We know the indole acid, right? So we have what are called plant growth regulators. In that plant growth regulators, we have auxin. So in that auxin, we have what is called the indole acetic acid. So indole acetic acid we have, yes. So here that is going to help. This is one of the natural, that auxin, right? So it is help in the synthesis of that auxin that is needed for the growth of the root and shoot. Yes. What about histidine? Yes, histidine, here one mistake is there. It is not removal of the amino group, rather it is decarboxylation we say. So what has to be removed there? It is a COH group that has to be removed. So let us see. So what is happening here? Please children. So what is happening here? See, look into this. Here in this histidine, you see the histidine decarboxylase. So what is happening? It is removing. So the carbon dioxide is removed. So through the decarboxylation, it is not the removal of the amino group, rather it is the decarboxylation. So that concept you have to know. So you have given the right answer, children. Excellent. Okay, very good. Now coming to maltose. Maltose is formed of 2 molecules of fructose, lactose, glucose and sucrose. So fructose, we know maltose, it's a disaccharide, right? So what is the monosaccharide when it is breaking down further? So suppose you take it in our intestine, we have maltase. So what will happen from the succus centricus? That maltase will break down the maltose to what molecules? So that should be there. So it's very simple and very direct, children. So what is that? Maltose, we know it's a disaccharide, so it gives 2 monomers of glucose. So we have fructose, it's a monosaccharide. When you take lactose, the lactose, we have what is the galactose and glucose. What about sucrose? Yes, the fruit sugar, it's a milk sugar. Fruit sugar, we have what is called glucose and fructose. Maltose, the 2 molecules of glucose. So what should be your answer? It is 2 molecules of glucose. So that concept you should know. So very good, children, you have given the right answer. Yes, excellent ma. Manjula, you have given the right answer. Very good, Manjula. Manjula, Jeevita, Lakshmanan, Akshaya, very good. You are giving the right answer. Excellent, students. Okay, now we are going to the next question. Out of the following elements, which is present in least quantity in human body? Like when you take the earth crust as well as the human body, the amount of elements that is quite distributed will be like quite more or less similar. But there are some elements which will be least distributed in the body. So we will see what are those elements: carbon, oxygen, silicon and nitrogen. So among this, which is least distributed? Yes, the answer is very simple. Like we know that carbon, oxygen, nitrogen, they are more vital, right? So what should be the answer? So you see carbon, it's around 18.5, oxygen 65%. What about nitrogen? 3.3. But which is negligible? It is the silicon. So the answer is silicon. Very good, students. You have given the right answer. Lakshmanan, Akshaya, you have given the right answer. Now coming to the metabolites. Okay, which one of the following statement is incorrect? Primary metabolites have indefinable functions. Some secondary metabolites have ecological importance. Secondary metabolites like rubber, drugs, spices and pigments are useful for human welfare. Secondary metabolites are not found in fungi, microbes and plants. So what should be your answer? So when we talk about these metabolites, you have to be very, very careful, because we know the primary metabolites, like what is that directly obtained. Secondary metabolites, we see they are obtained from the trace ones. So you see primary metabolites have indefinable functions. Okay, that's something correct. What about that they have ecological importance? Yes, the rubber, drug, spice, we are using it. As we saw, whatever the spices, what we get, even we get the digitalis, right, the drugs, okay, the cinchona, the quinine, the anti-malarial drug, they are all secondary metabolites only. And these secondary metabolites, they are not formed in fungi, microbes, and that cannot be accepted because they are formed. So that is the wrong option. They are found in fungi, microbes and plants. So that is the incorrect statement. Okay. Now coming to this answer, this question, ATP, adenosine triphosphate, ATP liberates high energy by breakdown of glycosidic bond, hydrogen bond, phosphate bond, both glycosidic and phosphate bond. So what should be your answer? We know ATP, yes, the currency of energy, the energy currency. Ultimately whatever is being taken, all are getting broken down, and those with the help of oxygen, they will be burnt. The fuel is burnt, and what is released in the form of 38 ATP molecules. So once when the ATP molecules are released, okay, how is that they are going to be going to liberate the high energy? So probably by the breakdown of what? Yes, your children, you are right, it is by the phosphate bond. See, you see the high level thing. So what happens when they go in for the reaction, they get broken down. So they are going to convert themselves into the adenosine diphosphate and inorganic phosphate, and that free energy is getting related. So what is the breakdown here? It is the phosphate group, not the other things. So that is the answer. Excellent, ma. All Jeevita, Akshaya, Lakshman and all, they have given the answers already. Very good. Next, coming to another question here. The enzymes catalyzing the breakdown of larger molecules into smaller molecules are called hydrolases, isomerases, ligases, both 1 and 3. See the enzymes, now we are talking about the enzymes. Some enzymes help in breaking down the bigger one to smaller one. What are they called? Now first we will see about ligase. Ligase is an enzyme which is used to link or join both the molecule. What about isomerases? Good example we can give, glucose and fructose. Yes, those isomers, they will be they will be having that stereomeric configuration, right? But they are the isomerases. They just make the exact mirror image or that optical L or D form of it. But what about the hydrolases? They are the one which will help in the breakdown. Particularly when you ask me to give the example, I will give the digestive enzymes. So what is happening there in the digestive enzymes? We are having what is called the hydrolysis. So here the larger molecules, here they are being broken down into the smaller molecules. So that concept is what is applied in the hydrolysis. So see here, it is going to help in catalyzing the breakdown of large molecule to small molecule with addition of water, the hydrolysis. So that you have to understand. Okay. But what about isomerases? There is rearrangement of molecular structure. Like I told, no, glucose, fructose, what happens? Okay, so that rearrangement will happen. But what about the ligases? They help in joining. So that is where, so what should be your right answer? Yes, it is the hydrolases. Excellent students, you have given the answer. Okay. Now this is a very simple question. If you know the Chargaff's rule, you can very well rock in this question, students. If the sequence of bases in one of the DNA strand is A, GGA, GAA, then the sequence of bases in other complementary strand of DNA would be CC, TT, CTT, TC, TC, TCC, TC, CT, CTT, CCT, CT, CT. So how are we going to make it up? So what should be your answer here, children? It is very simple. We all know the Chargaff's rule, right? I'm sorry, the Chargaff's rule. Okay. So in Chargaff's rule, like what we usually say. So in Chargaff's rule, okay, in Chargaff's rule, yes. So see, A is to T, okay, then G to C, G to C. So somewhere, so G to C. So this is not the one. So this cannot be, this cannot be. So G to C. Then what about A, T? Then G, C, A, T, T. So what should be your answer? Yes, students, it is none other than the 3, because that complementary linkage is what is formed. See, A means you just look into the option. So TT is coming. But what happens to the next thing? With the G, C is coming. But next to G, it is not coming, right? So this cannot be the option. So what it should be? It should be the third one. So that is the right option. Okay. So you are applying the complementary linkages base pairing rule and you are trying to match the complementary strand. Like this questions will come. If you know the concept, it's very simple and you can attempt also. Next, coming to this question, for its activity, carboxypeptidase. So we have enzymes, right? Carboxypeptidase, which comes from the pancreatic, exocrine pancreatic secretion. Yes, the carboxypeptidases. We have heard about it, which will be activated by the trypsin. It requires copper, zinc, iron and niacin. Carboxypeptidase is an exopeptidase. So it will cleave at the terminal ends and it will try to release the amino acids directly. So that is the beauty of carboxypeptidase. Usually they are secreted as pro-carboxypeptidase and only it is the trypsin which will make it into the active form of carboxypeptidase. So for the carboxypeptidase, for its activity, what is that cofactor that is needed? So what is the answer, ma? Yes, students, I am waiting for you. What should be the answer? Which enzyme is needed? Yes, you are right, it is zinc. So you can see here the zinc. So that is, so you can see the zinc is needed for it to act. Okay. So you should know that for carboxypeptidase, zinc is required for its activity. Next we are coming to the agar. Agar is commercially obtained from, actually you know agar, it's an unbranched polysaccharide, right? So it's an unbranched polysaccharide which is obtained from red algae, blue-green algae, brown algae and green algae. So from where it is obtained? Agar. So agar is obtained from, yes students, from where it is obtained? Yes, you are right, it is obtained from the red algae. So you can see that it is a type of mucopolysaccharide. Okay, it is obtained from the cell walls of the red algae, the gelidium and all we will say. So that is very much used in the what you call the preparation of that medium. Okay, as well as we will be using in all the isolation electrophoretic techniques and all, where we will be using the agar gel. And apart from that, we are using in cosmetic as well as in textile. Okay, everywhere this agar is being used. Okay, so agar, it is obtained from where? From the red algae. Very good children, you have given the right answer. Excellent. Next, very direct question, ma. Lecithin. Lecithin is a phospholipid, carbohydrate, protein, amino acid. So what should be your answer? Lecithin. It's definitely not a carbohydrate. It's not a protein, not an amino acid. So what should be your answer? It's a phospholipid. So this lecithin is something, it's present both in, see, you can see the compound here, right? So it's a phospholipid. See how it is attached. Okay, it is present both in plant and animal tissue. So that is where the lecithin has got its importance. Okay. So it's a phospholipid. Now coming to the last question, here they have given a list of substances that we are going to match with their functions: collagen, trypsin, insulin, receptor, GLUT4 and antibody. Here they have given hormone, enzyme, fight infectious agent, enable the glucose transport into cells, sensory reception, intercellular ground substance. So how are we going to match? Yes students, I am waiting for your answers. So when you see collagen, what should strike your mind? Collagen, trypsin, insulin, receptor, GLUT4, antibody. Antibody, can we take it like this? It will fight against the infectious agent, it fights the infectious agent. What about GLUT4? Enables the glucose transport. What about receptor? For sensory perception. Insulin, hormone, trypsin, enzyme, and collagen, the intercellular ground substance. Yes children, can we take it like that? Yes. Shall we proceed? See? Yes. Very good Akshaya, Jeevita, Lakshmanan. You are giving the right answers. Very good. See, when we say collagen, okay, this collagen already we saw those fibers, they form that intercellular ground substance, the extracellular matrix. What about the trypsin that has been secreted from the pancreatic juice, which is secreted in the inactive form and that is made active, okay, by the enterokinase, it is made active. And then the trypsin, which is a powerful proteolytic enzyme, it's a proteolytic enzyme, okay. And what about the insulin? It is well known, hypoglycemic hormone, right? And what about receptors? They are biological transducers. They have the capability to convert one form of energy to another form of energy. So we have that sensory perception like olfaction, smell, taste, etc. What are blood flow? They are glucose transporters. Okay, so they will help in transporting the glucose. More they are being distributed in the muscle cells as well as intestine and renal cells, where we see the GLUT4. Okay, the GLUT4, many work they have done. They say how exercise is reducing the blood glucose level. They will say that GLUT4, that glucose receptors, they will just get incorporated in the membrane, and without the assistance of insulin, they can help in allowing the glucose to be absorbed. That is where some new research they have made. And what about the antibody? It has got the ability to fight the whatever toxins or pathogens that is entering the body. So like that, yes. So this is where the answers that we saw. So I hope students, we had all this whole of this hour where we had a very good, nice interactive session with the 30 fast, interactive, rapid, quick revision on the session on the biomolecules. I hope it would have been very interesting as well as more informative. But I had a very good, wonderful interactive session, particularly Akshaya, Jeevita, Lakshmanan, okay, and Manjula, all that. They have been very interactive throughout. So thank you, students. Okay, thank you so much. I'll meet you shortly in another nice, interesting session shortly. I'm taking leave. Thank you, students.